Imagine holding the instruction manual for a human being, not just reading it, but rewriting and designing entirely new sections. Picture the power to edit, rearrange, and even invent the very code that makes us who we are. This is no longer the stuff of science fiction, it's the dawn of a new era in biology. As of early 2025, this vision is becoming reality in the UK with the launch of the Synthetic Human Genome Project, or SynHG. This groundbreaking initiative is set to transform our understanding of genetics and human biology. SynHG brings together the world's leading geneticists, biologists, computer scientists, and ethicists. Their mission? To build a complete, functional human genome entirely in the lab using the latest advances in synthetic biology and computational modeling. The goal isn't to create artificial humans but to construct human DNA from scratch. By doing so, scientists hope to unlock the secrets of every gene, including the mysterious stretches of so-called junk DNA that have puzzled researchers for decades. This ambitious project builds on decades of pioneering work such as the successful synthesis of simpler organisms like yeast and bacteria. Each step has brought us closer to understanding the complexity of human life at the molecular level. The UK government, along with international partners, is investing heavily in SynHG. They see it as a quest not just to revolutionize medicine, but to fundamentally reshape our understanding of what it means to be human. The project is a marathon, not a sprint. Over the next decade, scientists will painstakingly assemble all 23 pairs of human chromosomes, setting milestones along the way and pushing the boundaries of what's possible in genetic engineering. Why now? The answer lies in technology. DNA synthesis costs have plummeted, and revolutionary tools like CRISPR have advanced rapidly, making this once unthinkable project finally within reach. The convergence of these technologies has opened doors that were previously locked, yet even with powerful gene editing, we still don't fully grasp the ripple effects of our changes. Every tweak to the genome can have unexpected consequences, and the complexity of genetic interactions remains a vast frontier. SynHG aims to fill those gaps in our knowledge, creating a gold standard reference genome for future research. This reference will serve as a foundation for scientists worldwide, enabling new discoveries in health, disease, and evolution. By building DNA from the ground up, scientists can finally test their theories in a controlled, unprecedented way. This approach allows for precise experiments that were never possible before, offering insights into the very blueprint of life. The excitement is global. This is the next leap in understanding what it means to be alive. As SynHG unfolds, the world will be watching, eager to see how this bold experiment will shape the future of science, medicine, and humanity itself. Synthesizing a human genome isn't just about reading DNA, it's about actually writing it, letter by letter, as if composing a brand new book from scratch. This process is far more complex and creative than simply copying what already exists. The original Human Genome Project was like photocopying an encyclopedia. It gave us a reference copy of all the information, but didn't let us change or improve it. In contrast, SynHG is about typing out a new, improved edition from scratch, editing, updating, and even rewriting sections to see what happens. Scientists use the 4 DNA building blocks, A, T, C, and G, to assemble the genome in the lab, following the reference sequence, but with the freedom to make intentional changes along the way. This process lets them understand not just the story, but the structure, function, and subtle nuances of our genetic code, revealing layers of information that were previously hidden. By writing the genome, they can make deliberate changes, testing the function of mysterious or poorly understood DNA sequences, and even correcting errors or adding new features. The goal isn't just replication, but improvement. Removing repetitive or unnecessary sequences, adding unique watermarks for tracking, and customizing the genome for specific research needs. This makes the synthetic genome a powerful research tool, opening up possibilities for experiments that were never before possible. The synthetic genome becomes an annotated, experimental platform, impossible to achieve with natural DNA, and designed specifically for scientific discovery. Scientists can now test hypotheses about gene function and regulation directly, observing the effects of their edits in real time. The result is a designer genome, ready for experiments that could transform our understanding of biology, disease, and even evolution itself. Writing the genome is the key to unlocking its deepest secrets, and may one day allow us to rewrite the story of life itself. Synthesizing a human genome begins not in a petri dish, but on a computer, where scientists use powerful software to design and map out the entire genetic blueprint. The digital DNA sequence is carefully broken down into smaller, manageable chunks, each representing a section of the genome that can be physically constructed. In the lab, automated DNA synthesizers take over, building these fragments by adding one chemical letter at a time, following the digital instructions with incredible accuracy. Thousands of these tiny DNA pieces are painstakingly created, then scientists and machines work together to stitch them together, much like assembling a massive, intricate jigsaw puzzle. Special natural enzymes are used to help link these fragments into larger sections, gradually building up to form a complete chromosome, one step at a time. At every stage, each piece and connection is meticulously checked and verified, ensuring that no errors slip through and that the final product is flawless. The end result is a synthetic chromosome, chemically identical to its natural counterpart, but engineered with precision and tailored to exact specifications set by researchers. These lab-made chromosomes are free from random mutations that can occur in nature, making them ideal for controlled scientific studies. Scientists can even add special watermarks or unique sequences to these chromosomes, allowing them to track and study their creations with ease. The ambitious SynHG project is working to build all 23 pairs of human chromosomes using this method, aiming to recreate the entire human genome from scratch. Once assembled, these synthetic chromosomes are introduced into living cells, where they face the ultimate test, whether they can function and support life just like natural DNA. This remarkable process transforms digital code into the physical blueprint of life, bridging the gap between information and biology. The unprecedented precision and control offered by this approach are opening up entirely new frontiers in genetic research and biotechnology. Synthetic chromosomes are not just a scientific achievement, they are the foundation for the next era of biology, promising breakthroughs in medicine, agriculture, and beyond. Creating a synthetic chromosome is only the beginning. It must function inside a living cell. Scientists insert the synthetic DNA into lab-grown human cells, using advanced techniques to deliver it into the nucleus. The cell's machinery must accept and read the synthetic genome, producing proteins and behaving normally. Watermarks in the DNA allow researchers to track the synthetic genome's activity. The ultimate goal: replace all natural chromosomes with synthetic ones, creating a cell line entirely controlled by lab-made DNA. These cells serve as a biological sandbox, letting scientists test gene functions by adding, deleting, or rearranging DNA. Early experiments focus on single chromosomes in isolated cells, ensuring safety and control. The aim isn't to create synthetic organisms, but powerful research tools. This approach allows direct testing of genetic hypotheses in living systems. The process is cautious, methodical, and groundbreaking. Synthetic cell lines will transform how we study human biology. SynHG could finally reveal the true function of every part of our genome, including the mysterious junk DNA. By building genomes with specific non-coding regions removed, scientists can observe the effects and pinpoint their roles. This bottom-up approach creates a detailed functional map of our DNA. Synthetic genomes also let researchers study how gene organization and 3D folding affect cell development. Scientists can redesign chromosome layouts to uncover hidden principles of gene regulation. Comparing synthetic human genomes to those of other species will clarify what makes us uniquely human. Experiments with Neanderthal or chimpanzee gene variants could provide direct evidence for theories of human evolution. SynHG is the key to decoding the blueprint of life. SynHG's practical promise is immense, especially for medicine. Synthetic genomes let researchers model complex diseases by introducing specific genetic variants and observing real-time effects. This could revolutionize drug discovery, enabling personalized treatments tailored to an individual's unique DNA. Synthetic cells could also help create rejection-proof organs for transplantation, ending long waiting lists. In the future, synthetic chromosomes might deliver healthy genes to cure genetic disorders at their root. These advances move us from trial-and-error medicine to precision-guided therapies. The groundwork laid by SynHG could make once unthinkable treatments possible. The project's impact will extend far beyond the lab. SynHG is paving the way for a new era in healthcare. With SynHG's power comes profound ethical questions. The project is strictly limited to lab-grown cells, not creating synthetic humans, but the technology could be misused. Where do we draw the line between therapy and enhancement? What does it mean to create life from scratch? The project's leaders are engaging ethicists, policymakers, and the public to address these issues now. Data ownership, biosecurity, and equitable access are major concerns. Safeguards and transparency are essential to prevent misuse and ensure fair distribution of benefits. The ethical framework must evolve alongside the science. These questions are as important as the research itself. SynHG's leaders know public trust is essential, so they've made engagement a core priority. Nationwide outreach, town halls, exhibits, online forums, invites everyone into the conversation. The goal isn't to sell the project, but to co-create a responsible path forward with society. Diverse voices, students, patients, faith leaders, are shaping the project's ethical guidelines. Scientists can explain what's possible, but society must decide what's acceptable. The future of synthetic biology will be shaped in labs and in public debate. Open, inclusive dialogue builds the foundation for trust and progress. Together, we decide how to use this new power. In 2025, we stand at the threshold of a new era, one as transformative as the dawn of computers or space exploration. SynHG is more than science, it's a catalyst for reimagining biology, medicine, and our relationship with life itself. Soon, writing DNA may be as routine as reading it, unlocking innovations we can barely imagine. We could see biocomputers, pollution-eating microbes, and new materials, all built from synthetic genomes. The line between natural and artificial will blur, forcing us to rethink our place in the universe. SynHG holds up a mirror to humanity, challenging us to decide what we value and what we wish to change. The journey will be long, with setbacks and breakthroughs, but it marks a turning point. We are becoming the authors of our own genetic code. With wisdom and humility, this power could heal, inspire, and transform our world. The next chapter of life is ours to write.